Welcome to our third video on topic 7 of our advanced higher level biology. We're in nucleic acids. We are looking in 7.3 at the process of translation. Okay, so remembering that translation is this process that occurs on the ribosomes, and it's where our genetic message as such that's carried in our mRNA is now built into a protein, okay, a functioning protein. So let's look at that in a bit more depth. The process of translation is whereby our codons, so those three groups of 3 bases on the mRNA, which were produced during that process of transcription, are translated now into our amino acid sequence using ribosomes and tRNA. So as we said, it occurs at the ribosome. Translation can only occur immediately after transcription in prokaryotes due to the absence of a nuclear membrane. So because it doesn't have to get out of that membrane, it occurs immediately to translation, then transcription. Whereas in our eukaryotes, we've got to get it out of the nuclear pore, out into the cytoplasm, and find in the ribosome. So it takes a little more time in eukaryotes. Okay, so let's think about ribosomes then. These are the site of protein synthesis. We've got 70 S ribosomes in prokaryotes, and we have 80 S in our eukaryotes. They consist of a large subunit and a small subunit. Okay, so they're made up of rRNA, so that's ribosomal RNA, and protein. They're free in the cytoplasm, or they can be bound to ER, forming what we call rough ER. Translation occurs in the space between the 2 subunits. So you see how we said we've got that large subunit and the smaller subunit. Transcription occurs in the space between the 2. We've got 3 binding sites for the tRNA. So tRNA coming in with its amino acid. There's 3 different binding spaces in here. We've got A, P, and E. So you've got binding site A, binding site P, and binding site E. Okay, so looking a bit closer at what we mean about these binding sites. So binding site A holds the tRNA carrying the amino acid that comes next into the polypeptide chain. So here it comes in and A is going to hold it. P holds the tRNA carrying the polypeptide chain. So P has the chain on it. And then E is the one that ejects the tRNA that is donated to the amino acid chain. All right, so let's have a look at our picture here. We've got our tRNA coming in. It's got our amino acid on the end. Okay, in it comes. Here in P, this one has got the amino acid chain on. As you can see, it's only got 1 amino acid on our chain so far. Then that moves to E, and E ejects it off out. Alrighty, so to think in this way, we need to think about our tRNA molecule. Okay, and an amino acid is loaded into the tRNA molecule using ATP and a specific enzyme. So it does require energy for this to occur. We've talked about that. Each amino acid has its own enzyme. 20 amino acids, 20 different enzymes. When the amino acid is attached, the tRNA is said to be activated. The enzymes are called tRNA activating enzymes. So to activate it, they attach their amino acid. So how does this happen? So the removal of phosphate from the ATP molecule, phosphorylation as we know, provides the energy required to load the amino acid to the tRNA molecule in order to reactivate it. Enzymes are specific. This enzyme will only catalyze the activation of one specific tRNA molecule, therefore loading one amino acid. So that's how comes these are specific to each amino acid. Okay, phases of translation. So we've always looked at translation, but we've not looked at it that deeply. Now there's actually various phases as we go through the process. Now we've got 4 phases. We've got initiation, elongation, translocation, and then termination. Okay, and some of these phases sound quite familiar to our PCR.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Alrighty, so we've talked about those phases of translation. Now, that video highlighted it really well, but let's look a little bit deeper. So we start with initiation. An activated tRNA molecule with methionine combines with the mRNA strand. Okay, so our start codon is our UAC or our AUG, all right, and methionine is our first amino acid. That's that starter. The subunit of the ribosome slides down the mRNA molecule until, so we're talking the small subunit, so that's going along the bottom, until it comes into contact with the tRNA, okay? So the tRNA met. The large subunit of the ribosome then joins the small subunits. They kind of connect over it. Now we work onto elongation. So elongation of the polypeptide chain. So that's the production of that polypeptide chain. And this involves those condensation reactions between the amino acids. Our activated tRNA molecules bring amino acids to the ribosome in the specific order the codons and the mRNA requires. The ribosomes help form those polypeptide bonds. This occurs at site P. Okay, next phase is translocation. Okay, and this is happening at the same time as elongation. Our tRNA moves from A to P to E sites. Okay, so A, the polypeptide chain is attached to the tRNA. P, the tRNA transfers the polypeptide chain to the next tRNA, which is now on the A site, and at E, the tRNA is then ejected from the ribosome to go and get its new amino acid. Then we get termination, and this is where a stop codon shows up on the A site. This causes a protein called a release factor to fill the A site. The release factor then causes the ribosome to separate and releases the mRNA and polypeptide chain. This is the end of translation. Okay, just taking a back step, remembering that part of your standard level course was that you know how to use an amino acid or a codon table. It could be in any form. There's various forms of these that get out and about, and in exams they do like to confuse you with a nice different format, so just be prepared to be able to read them. All right, so what happens once our polypeptide is formed? What's next? So they can go to various destinations. Usually polypeptides are made of on the free ribosomes. They are for use anywhere within the cell. Polypeptides made on rough ER are secreted from the cell or used by our lysosomes. So now we're going to look at that protein structure a bit further. You guys should already know the stages of your protein structures. Okay, so going from that polypeptide chain, being the primary structure, through to the secondary structures, which can either be our alpha helix or the beta shape, and then moving on to the tertiary protein structure, and then that quaternary, sometimes where several proteins interact. Okay, so looking at the primary structure first of all, this is our unique sequence of amino acids. They determine the structure of the other three levels, because depending on the organisation of those amino acids depends whether the bonds are going to form for the protein to develop. Each amino acid is held together by a peptide bond. Alterations, mutations, can have a drastic effect on the protein function, as the shape of the protein equals the function. So if you change that amino acid chain, the shape of the protein will be different, therefore the function will be impacted. Okay, with the secondary structure, we said we've got this alpha helix or the beta shape, which is the beta pleated sheet. So these are created by the hydrogen bonds forming between the carboxyl group of one amino acid and a hydrogen group of the other amino acid. It is not related to the R group. So secondary structure is carboxyl group and hydrogen group interactions. When we move on to the tertiary structure, this is what we call those globular protein shapes now. This does involve the R groups. Okay, so the polypeptide chain now bends and folds due to interactions with those R groups. So you can see how it's all quite intrinsic, but it really depends on that amino acid change, that amino acid chain and organisation. Now, due to the interaction of these R groups, this can be due to polarity, hydrophobicity, charge, anything along those lines, okay? And you must remember, we're thinking of this as a 3D shape. This is not just 2D. Now, in some cases, our polypeptide chains interact with other polypeptide chains, and you end up with what we call the quaternary structure. Now, this isn't always present, so it doesn't always happen. You don't always have proteins that are several chains interacting, but sometimes you do, okay? Alrighty, so we've looked at that protein structure, which is great. We're just going to move on to something now called polysomes. And the idea of polysomes is that when you get a section of mRNA that's being read by a ribosome, we've got it in our mind that our one ribosome moves along our mRNA and makes our polypeptide chain. Now, that's true. However, biology is much more efficient than that. So you get these things called polysomes, and this is where we have several ribosomes reading an mRNA strand. So what it means is that we don't just get one length of mRNA, it's read, off it goes. We can make several polypeptide chains from one piece of mRNA if we have several ribosomes reading it at a time. Okay, and we call that polysomes, because when you look at this, the micrograph image, you can see that it looks like a bead of beads on a string. Okay, and that's because there's several ribosomes working there, creating their own polypeptide chains. Now, this can occur in prokaryotic cells and eukaryotic cells, both on the free and those bound ribosomes. And what it means is it's possible to make a lot of a certain polypeptide from one single mRNA strand. And that is us done on 7.3, translation. I'll see you in our next video.